Assalamualaikum and good morning. Today we are going to learn about cell suspension culture. What is cell suspension culture? Suspension culture is a type of culture in which single cells or small aggregates of cells multiply while suspended in agitated liquid medium. It is also referred to as cell culture or cell suspension culture. Establishment of single-cell cultures provide an excellent opportunity to investigate the properties and potentialities of plant cells. Such system contributes to the understanding of the interrelationships and complementary influences of cells in multicellular organisms.
To initiate single-cell cultures, batch culture is used to provide suitable environmental condition in a phase among culture medium, while continuous culture, fresh medium is continuously added and metabolic end products are removed. The usage of cell suspension in plant biotechnology includes mutagenesis, embryogenesis, protoplast, gene transfer, and production of secondary metabolites. Hello, now we're going to explore the application of cell suspension culture, which is in plant mutagenesis. Before that, do you know what is mutagenesis or mutant? Not this guy, okay? Well, mutagenesis is a process that introduces agents that can cause mutation to plant DNA. The changes are highly specific too. Mutant, on the other hand, is a product of mutagenesis which display distinct feature or resistance towards certain chemicals compared to the original plant due to the result of DNA alteration. Generally, there are two types of mutation, chemical and physical mutation. Basically, before being treated with any mutation, plant cells will be selected and suspended in broth media that will proceed to the steps of mutagenesis. Commonly, chemical mutation uses alkylating agents such as MNNG, N-ethyl N-nitrosourea, and ethyl methane sulfonate. As shown in figure right here, Nippon bear seeds was treated with EMS. When the plant successfully grew, M2 generation plant was taken for genetic analysis to observe the mutated gene that have been passed down caused by the first generation of mutated plant cell. Next, physical mutagens. These agents such as X-rays, UV rays, and gamma rays are commonly used in plant tissue cultures. The cell in the suspension culture will be treated with X-rays or gamma rays to break the DNA strand, whereas UV rays increases the occurrence of pyrimidine dimers in the DNA sequence. The cell will grow in vitro and undergo further analysis evaluation. The beautiful pink flower here is belong to the Nicotiana tobacco plant. X-ray treatment towards the Nicotiana tobacco cell lines promote the plant to resist paraquat herbicide, which is widely used to kill weed and grass. Now we know how the alteration of plant genetics was conducted, but why mutant plant cell culture are significantly useful in the study of the physiology as well as the improvement of crops? Well, with the help of genetic alteration, plants can carry new desired traits such as long shelf life, physically appealing, tastier, and many more. Due to this reason, our plant industry can grow globally, hence able to contribute economically.
Now we are going to move to the embryogenesis. Plant embryogenesis is the mechanism whereby embryos of plant grow and develop. Through embryogenesis, the basic body plan of the plant is developed. The primary plant tissue types are differentiated and the food reserve accumulated, which will eventually be used by the germinating seedling after a period of dormancy. In addition, embryogenesis is also the method of the unicellular fertilized zygote which make a progressive transformation to the embryo. Through embryogenesis, two stem cell populations are formed, which are the root apical meristem that known as ram, which provide the meristematic cell for the future root growth. The second one is shoot apical meristem, which is sap, that give rise to organs like the leaf and the flower. These two meristems will generate the entire adult plant morphology. For the root apical meristem and the shoot apical meristem, you can refer this figure. The main body part of the matured embryo include first apical meristem, second hyposotile, which is the embryonic stem, third cotyledon, which is the embryonic leaf, and the last one is roots and roots meristem. There is the radicle, which also known as embryonic root, whereby the radicle is the first part of a growing plant embryo. For the main body part of matured embryo, you can refer this figure. Artificial seeds are one of under the embryogenesis suspension cell culture. Artificial seed can be defined as the growing seed-like structure that are experimental study which developed by a process in which somatic embryo extracted from plant tissue culture then become encapsulated by a hydrogel. After that, the encapsulated embryo serve as true seed once grown in the soil. It can also be used as a replacement for natural seed. This figure shows the artificial seed. This is the general method for making artificial seed. First, the establishment of callus culture. Second is the induction of somatic embryogenesis in callus culture. Third is the maturation of somatic embryos. And the last one is the encapsulation of somatic embryos. This figure shows the steps for the synthetic or artificial seed production, which are from the explant, that is leaf, stem, root, and etc. can become the artificial seed. These are the advantages and disadvantages of artificial seeds. The advantages of artificial seed are: first, it can be done in a large-scale propagation. Second, variety of genotypic plantation can be obtained. Third, important source of germplasm conservation of superior, extinct, and endangered species can be preserved. And the last one is it really easy to handle transport, and also the transport charges are very low due to their small sizes. For the disadvantages are: first, it is really expensive due to this facility. Second, it requires a special skill to operate the artificial seed production.
What is protoplast? Protoplasts are plant cells with a plasma membrane but without cell wall. Because of this, the protoplast provide a starting point for many of the techniques of genetic manipulator of plants, in particular the induction of somaclonal variation, somatic hybridization, and genetic transfer. Protoplast culture. Protoplast culture is the aseptic isolation and culture of protoplast in vitro so as to obtain a viable plant. Isolation of protoplast is by two methods. The first method is mechanical method and the second one is enzymatic method. Protoplast purification. Enzyme treatment results in suspension of protoplast, undigested tissues, and cellular debris. This suspension is passed through a metal sieve or a nylon mesh in order to remove undigested cellular clumps for protoplast purification. Culture of protoplast can be done by two methods. The first method is feeder layer technique and the second one is co-culture technique. For the feeder layer technique, the protoplast plated into an agarified medium are exposed to X-ray dose. In the co-culture technique, protoplasts of two different species are cultured together for promotion of growth. Regeneration of protoplasts can be divided by two, which are formation of the cell wall and the formation of callus and platelets. formation of the cell wall show that regeneration of new cell wall over the plasmilla, and the formation of callus and platelets show that after the first division of the cultured protoplasm, subsequent division yields small cell colonies on suitable semi-solid medium. Examples of application of protoplast culture. First, protoplast can be used to study membrane biology. 2. Use isomaclonal variation. 3. DNA transformation. And the fourth one is plant breeding using a technique called protoplast fusion. Cell modification. What is cell modification? Cell modification is the most recent aspect of plant cell and tissue culture that provides the means of transfer of genes with desirable traits into those plants and recovery of transgenic plants. The technique has a great potential of genetic improvement of various group plants by integrating in plant biotechnology and breeding programs. It has a promising role for the introduction of agronomically important traits such as increased yield, better quality, and enhanced resistance to pests and disease.
What is gene transfer? Gene transfer is the transfer process of a gene of interest that is commonly a foreign gene ligated to the bacterial plasmid into the genome of the plant cells. Gene transfer are mainly divided into two types of method, which is direct gene transfer and indirect gene transfer. Direct gene transfer, or also known as vectorless method, involve the introduction of exogenous DNA directly into plant genome through physical or chemical reaction without use of any host, viral or bacterial system. Cell fusion or protoplast fusion is the example of direct gene transfer. Cell fusion can be spontaneous fusion or induced fusion. Indirect gene transfer, it is called vector-mediated method. It involves the introduction of exogenous DNA into the plant genome via biological vectors such as bacteria plasmid. Agrobacterium-mediated method is one of indirect gene transfer where TI plasmid with desired DNA, or also known as tDNA, is inserted into plant genome, producing plant with desired genetic traits. The diagram shows the overview steps of protoplast fusion. It starts with isolation of protoplasms, fusion of protoplasts from different genome by spontaneous fusion or induced fusion, selection of hybrid cells, and culture of hybrid cells which produce hybrid plants. On the other hand, this is the overview steps of agrobacterium-mediated gene transfer. First step is bacterial colonization, following with insertion of gene tDNA into TI plasmid, and the third step is introduction of recombinant gene into plant cell in the culture, and finally regeneration of plant with new trait. Gene transfer is a good method because of the diverse techniques that are available. The plant cells produced are totipotent, and we can obtain improved organisms. We also can reduce the chance of getting undesirable organisms, and we can save time as we produce many organisms at once. Unfortunately, gene transfer method is costly because the requirement of special equipment. Gene transfer method also must be performed in controlled conditions in the lab to prevent contamination. The regulation and stability of introduced genes is also still variable, and there is also ethical issue that arise from this transfer method.
Secondary metabolite is a compound that are not required for the growth or reproduction of an organism but are produced to confer a selective advantage to the organism. Why secondary metabolite? First, protect plant from the attack of predator, pathogen or competition. Second, important for abiotic stress. Third, medicine for industrial activities. There are four classifications of secondary metabolite. First, chemical structure. Second, chemical composition. Third, solubility in various solvents. Fourth, pathway by which they are synthesized. Types of secondary metabolite. First is alkaloid. It's derived from amino acids such as ornithine, arginine, and lysine. It has ring structure and a nitrogen residue, and wide use in medicine. There are two uses of alkaloid in plant. First, poisonous agent which protect plants against insect and herbivore. Second, for regulatory growth factor. Phenolic. First, it derived from aromatic amino acids such as phenylalanine, tyrosine, and tryptophan. Second, it resides in every plant that they attract pollinator to the plant and impact how these plants act with one another. Flavonoid. It gives plant red, violet, or blue color. Second, inhibit the growth of bacteria and viruses. And third, protect the cell against the damage of free radical. Sulfate. It contains sulfur. Second, it belongs to lily family onions, leeks, asparagus, and garlic. Third, it can lower cholesterol level. And fourth, preventive effect against cancer. Curcumin. First is extract from turmeric. Second, it can be used for coloring and medical uses. Third, anti-ulcer. And fourth, antimicrobial and antiviral.